I will be talking about physical models of biological machines. And as a biophysicist, I'm really fascinated with the ways that we could use both the concepts and the problem-solving tools of physics to help answer interesting questions in biology. So I'll talk about some biophysical problems I'm interested in on two different scales. So on one scale, whoops, getting the hang of this laser pointer. There we go. On one scale, I'm interested in how things move inside of cells. So for example, during mitosis, chromosomes line up at the center and then need to be segregated. So there's some interesting physics questions to do with what actually produces that motion. How are they segregated? Another area on this scale would be how are vesicles transported through cells to specific destinations. So this cartoon here shows a nerve cell with a long axon and the nerve terminals, and material needs to be able to move all the way from the cell body out to the terminal, and something needs to move things back from the terminal back to the cell body. So an interesting physics question is to try and understand what produces that movement. So that's kind of on the molecular scale inside of cells. I'm also interested in forces and movements happening on the cell scale. So how do biological cells perform mechanical tasks? So one example would be during cell division, a cell needs to actually physically divide. So this is an image that shows a contractile ring, which is a bundle of filaments that forms around the perimeter of a cell, and then this bundle of filaments contracts and actually pinches the cell off into two separate daughter cells. And so there are a lot of physical questions in understanding how that whole process happens. Another example is cell motility. We don't always think of cells as things that move in themselves, but movement of cells is really important for a lot of processes. For example, a white blood cell needs to actually chase an infectious bacteria in order to catch it as part of our immune response system. Another example would be if you have a wound in your skin. If you get a cut, the surrounding skin cells actually move in toward the site of the wound in order to repair it. So cell movement is a really important biological process. So one really important system underlying a huge range of forces and motion within cells is the cytoskeleton. So this is a dynamic network of polymers that provides kind of a mechanical scaffolding. And this is an image that shows some of these polymers. The fluorescently green marked polymers are called microtubules, and the red fluorescently marked polymers are called actin. And what's marked fluorescently in blue is actually the nucleus of this cell. So the cytoskeleton has several mechanical roles. One is that it provides mechanical structure. So this is why it's actually called a skeleton, right? It actually provides a mechanical framework so that the cell can withstand mechanical loads. Another thing that it does is it supports cell movement and cell division. So this framework of polymers, unlike our skeleton, can rearrange very rapidly. And as it rearranges its structure, that is what allows the cell to change shapes rapidly and do things like divide or change shapes and turn corners. So we need a dynamic cytoskeleton in order for the cell to move and change its shape. And another really important role for the cytoskeleton is that this interconnecting system of polymers also happens to provide a system of roads. So for material to be transported inside of the cell, we actually have a whole bunch of little tracks that things could be transported along. I want to take a minute to think about that last role of the cytoskeleton that I just told you about, that it can function as a system of roads. So we could actually think of it as sort of analogous to a city. And in this cartoon, what we have are some very long roads that go long distances. These would be analogous to the microtubules. And so these can be thought of as sort of the freeways of the cell for things that need to be transported long distances on the cell scale. We also have some short local roads, particularly toward the periphery of the cell. These are the actin filaments. For things that need to be transported shorter distances, often this happens along actin filaments. And then we have some molecules called molecular motors that are kind of analogous to the cargo trucks in this situation. So materials that need to be moved around the cell are packaged into these kind of packages called vesicles and carried along the filaments by molecules that are designed to move towards specific directions along these filaments. So this is a schematic of a few of the different types of molecular motors that serve as sort of the cargo trucks for transporting materials through a cell. So this one, called kinesin, moves cargoes toward the plus end of microtubules. And this other molecule called dynein also interacts with microtubules, but it moves toward the opposite end, what's known as the minus end. So we have one type of motor that's specialized toward moving things toward the plus end, which tends to be oriented toward the periphery of the cell, and one type of motor that's more specialized to move things toward the minus end, which tends to be toward the center of the cell. And then this is another kind of molecular motor that is specialized to carry cargo along actin filaments. And some features that they all have in common that I'll point out are down here, what are called head domains. And these are binding domains that can attach and detach to the filament that the motor is walking along. And as they do this, as they attach and detach, they can exert force with respect to the filament. So molecular motors can work in two ways. They can work individually. A single motor could actually drag a cargo along a filament over a considerable distance. Or in some cases, molecular motors work in teams, where we'll have a bundle of motors that work together to grab onto filaments that are nearby and pull on them locally. And when you have clusters of motors doing this, they exert a contractile force. And that's what's happening in, for example, muscle contraction. So I want to talk a little bit more about the mechanism of single molecule molecular motor function. So there are a wide variety of experiments that look at these individual motors and how they move along a filament in order to transport material. And one example is this one here, shown in the schematic, where a quantum dot is attached to one of the legs of the motor, and then they can monitor the position of this quantum dot with a very high degree of spatial and temporal resolution. And this allows them to, by proxy, monitor the position of the motor as it's moving along the filament. And one thing that's seen in the big range of single molecule experiments is that these motors move along in discrete stepwise movement. This is two different examples of this particular experiment shown on this position versus time graph. And when they look very closely at the spacing of these steps, another thing that they can deduce is that it's actually moving in alternating steps, in what's sometimes referred as hand-over-hand motion. They're taking alternating steps just the way we do. And so, for me as a physicist, some of the questions that really stand out to me about this movement is, one, what is the role of Brownian motion in the stepping mechanism? So these molecules exist in an environment where they are very small and they are surrounded by large water molecules that are moving around through thermal motion and bombarding them from every which direction. And so the motor cannot just walk really steadily like we would. It has, constant forces hitting it every which way, so its movement is going to be very thermally noisy, like what you can see in this movie. And so a big question is, how does it contend with that type of thermal motion? Is this a problem for it to overcome? Is there some way that it can make use of this motion to actually help with its stepping mechanism? So that's one area that I was interested in. The other is, yeah, sometimes the one in front actually detaches first. And that gets to my second question, how are the steps coordinated? How does the foot that's in front know it's in front? How does the one that's behind know that it's my turn to take a step, right? So that's kind of another big area where physics and biochemistry overlap, where we want to understand the coordination mechanism for that stepping. And as you can imagine, if it gets out of sync, you could have one of them detach, and before it actually reaches its new binding site, the other one detach, and then the whole thing diffuses away. And this happens in both the actual system and in the simulations, that eventually it may get out of sync, and so there's a specific number of steps that it might take before it diffuses away. So in order to get at some of those physics questions that I was talking about to do with molecular motor transport, we developed a mathematical model for myosin 5 based on some of the things we know about the physical properties of the parts. So we used information from single molecule experiments, crystal structures, and electron microscopy images in order to develop sort of a coarse-grained model where we put in what we thought were maybe the essential physical ingredients. So some of the basic assumptions that we made with this model are, one, we assume that these neck domains are semi-flexible. So in other words, they can bend, but there's some resistance to bending. I also assumed that these head domains bind and unbind stochastically with certain probabilities, and the probability for unbinding is going to be strain dependent. And what that means is there is a way for these two heads to communicate with each other because there is strain in the molecule. And so if the one that's in front experiences a strain backward, and this slows down its chemical cycle and makes it less likely to detach from the molecule, this could provide a coordination mechanism by which the motor might stay in sync. And the third really important assumption of the model was that when a head detaches, it's going to undergo Brownian diffusion until it finds a new binding site, until it gets really close, and then it can reattach. So we wanted to put these basic assumptions into a model and test it and see does it do what we hope it will. And so the tool that we'll use to test the performance of this model is Brownian dynamic simulations. So I want to give you just a brief overview about how this tool works. So Brownian dynamic simulations, the approach is as follows. I divide the molecule up into discrete points. You can see these kind of black points along the molecule. And I'm going to keep track of the position of those points. So for each point, I want to think about what are all the forces acting on that object, right? And so those of you who have taken physics may recall that Newton's second law of motion tells us that the mass times the acceleration is equal to the sum of forces on an object, right? But these molecules exist in a regime where there is so much viscous drag that the acceleration of the object is actually going to be damped out on the time scale that we're interested in. And this is what's referred to as overdamped motion. The way I include that in the simulation is that I say mass times acceleration is approximately equal to 0 on the time scale that we are interested in. And so what that leaves us with is an equation that says that the sum of all the forces on the object is equal to 0. And this is what's known as an overdamped Langevin equation. The reason that this equation is useful is that it provides us an effective equation of motion for each of the points in the object. So let's take a look at the forces that I chose to include in this equation. So the first one is a viscous drag force. So as the object moves through a fluid, there is the force of the fluid pushing back on it. The second force is an elastic bending force. So if we bend at any of those joints, there's going to be a restoring force trying to bring it back to equilibrium, to straighten it out. And then the third type of force that I include in the simulation is a thermal noise force. And this is to simulate the effect of those water molecules that are hitting the object from all different directions. And so we use a random number generator to generate that thermal noise force. So I said that this is an effective equation of motion, but how do we use it to actually simulate the movement of the molecule? What we want to do is solve this equation for the position of the object. And this is something that we can't solve analytically, so we use a computer to do it. And this is a discretized solution to this equation. And what it basically the approach is, I think about time in little discrete intervals, delta t, very small intervals of time. And I say, at my starting time t, I know the position of my object. And after a very small time, delta t, I'm going to update that position based on what I know about all the forces acting on that object. And so then that gives me a new position at time t plus delta t. So I perform that calculation for each object. Then I update the forces experienced by each of the objects. And if I do that many times over many, many delta t's, what I'm actually doing is simulating the movement of the molecule as a function of time. We actually see what the predicted movement would be. Oops. And so putting all those things together, just to remind you, I put in some assumptions about the head domains, the net domains, and the filaments, and a few simple rules that the head can detach, and the detachment probability depends on the force it feels, and it can reattach once it reaches the new binding site. And putting those things together, it should be playing the movie that you saw already, but when you put all those things together, what we get is that the motor actually undergoes alternating steps, hand-over-hand motion, and it reproduces some of the features that are seen experimentally, that it can take alternating steps, it can take many steps along the filament before detaching, and when we put in proper choices of parameters based on experiments, we also reproduce many of the features such as the velocity of the motor and the load dependence of the motor that are seen experimentally. So some future applications for this project could include extending this model to look at how does motor transport work in more crowded environments like what we see in the cell? Or what about when the motor reaches a filament intersection? What determines whether it's going to keep going on its original path or take a turn, for example? So a new direction that I've gotten very interested in recently is to look at molecular motor-based transport in axons. So this cartoon, what we see is a nerve cell, and then this is an axon. That's the channel along which signals are sent out from a nerve cell to a terminal. And within axons, there's a dense bundle of microtubule filaments that makes kind of a system of roads for things to be transported within the axon. And they are all oriented in such a way that their plus end is toward the outside and their minus end is toward the cell body. And this is important because that allows certain types of motors, kinesins for example, to carry cargo toward the outside. And it allows other types of motors, for example a dynein, to carry things all the way back to the cell body. And so for this type of transport to happen in a predictable way and to happen the way it's supposed to, we need this road actually set up so that it's polar, so that we have the plus ends all toward the outside. And as it happens, molecular motors are also involved in building this road. They carry very small pieces of microtubule along the already established bundle and incorporate these small pieces of microtubule into the bundle. So they play an essential role in establishing and maintaining a polarized microtubule bundle. this is important because when it goes wrong, it's associated with some neurodegenerative diseases such as Alzheimer's disease. This is a cartoon schematic of a disease scenario where we have a microtubule bundle where some of the microtubules are oriented the way they should be, and others are oriented with their plus ends inward. And so then the motors that are trying to carry things to the proper destination at the proper time are not doing what they should. So to try and understand the system and the mechanism for motor-based transport in establishing the filament bundle, I've been having some very fruitful discussions with these experimentalists who have developed ways of imaging the movements of the small microtubules. So this is a schematic of one of the experiments they do to visualize the movement along axons. And it's called FRAC, or fluorescence recovery after photobleaching. So what they do is they fluorescently mark all the microtubules. But with so many microtubules in there, all fluorescently marked, you can't really pick out the motion of any individual one. So next, they photobleach a region of the axon, and then they wait. And a small microtubule in this region that gets carried into this region is now visible, and they can track its motion. And this is an example of one of their early movies, where, maybe a little hard to see in this light, but we're going to see a small microtubule come into this region and be transported along. And this is an example of some data from one of their movies where they tracked the position of the microtubule as a function of time. And what we can see in this graph is that it looks pretty different from the discrete stepwise motion that we saw for an object being carried by a single molecular motor. We don't see discrete steps here. We see it move, slow down, kind of rock back and forth a little, move quickly, move more slowly, pause for a little while, move again, pause for a little while. There's more going on here than just one molecular motor carrying something in stepwise motion, right? I don't know if this movie actually played. Yeah, we can see. There it is. So there's a small fluorescent microtubule that we can see kind of moving along, jittering back and forth, and moving further along the axon. And this is at the early stage of Anand's experimental project. He's a graduate student who is developing ways to fluorescently track these microtubules at higher spatial and temporal resolution. So this was one of his early movies, and he's working at generating more of these so we can get a lot more data like this. And what I'm hoping to do is to develop models that will help us interpret what's going on underlying this motion. So what we think may be happening is that the microtubules are being carried by more than one motor, that multiple motors of different kinds combine to it at once. So you can envision what might happen if motors that want to go toward the plus end and motors that want to go toward the minus end all attach at the same time. You could get a situation where they're pulling in opposite directions, so it's kind of like a tug of war. So let's take a minute to kind of think about the physics of a tug of war. So let's say I'm having a tug of war here, and those of you who have taken physics may remember that Newton's third law of motion says that for every force, there is an equal and opposite force. So my opponent over here is pulling just as hard as I am. How does anybody ever win a tug of war, right? The answer is, it depends on how much traction we have with the ground, right? So I'm pulling, the other person is pulling just as hard, but if I have enough traction between my feet and the floor that I can withstand that pulling force, but my opponent's feet start to slip, that's when I can win, right? So we think that the same thing goes on the molecular scale. So if we have Team Diamine pulling this way, Team Kinesin pulling this way, but each of these molecules is kind of sharing the load, what happens if one of them detaches? One kinesin detaches, and now the remaining two kinesins are sharing that same amount of load, so they each individually feel a stronger force than they did before. And the more force they feel, the more likely they are to detach. Their binding affinity to the filament becomes weaker. So when one detaches, the remaining feel a stronger force individually and are more likely to detach also. So if another one detaches, then that remaining one is even more likely to detach. So what you can get is kind of a cascade of detachment, and then team diamine has won and can just take off in the direction it wants to go. Right? So I wanted to simulate this scenario, taking a look at a microtubule where different types of motors can attach and detach with certain probabilities. So the way that we do this, or the way that I approached it, was to say that we can have minus n-directed motors like diamine and plus n-directed motors like kinesin, and we want to simulate the movement of one microtubule. So we say at any given time, new motors can attach with some certain probability, and motors that are already attached could detach with some probability, and that probability of detachment will depend on the force that those motors feel. And so, just like in the last project, I divided up into discrete small time steps, and at every time step, I want to update the number of attached motors. So for that microtubule, I will perform this calculation to update how many kinesins and how many diamines are attached. And then when I have my new number of attached motors, then I can update the position of the microtubule based on basically which team is winning. Right? And if I do that over many, many time steps, I'm going to get a simulation of that experiment where we watch the movement of the microtubule. So this is a sample of some of my simulated data based on that model that I just showed you. So this is a position versus time graph of the microtubule, and we can see here it's going slowly, slowing down, now it's speeding up, now it's kind of paused. So we can see kind of different characteristic features of motion, kind of like what we might see in Anand's movies from the experiment. And with the simulations, we can also look under the hood and we can think about what's happening with the motors underlying this positional movement. So this is for that same simulation, looking at the number of kinesins and the number of diamines as a function of time. So you can see both of those numbers is fluctuating rapidly, and at any given time, like say here, we have 10 to 20 of one type of motor attached and a smaller number of the other. And it might switch places, where one team's winning and then it switches. And in here we can see there's actually about the same number for a little while. And let's look up on the position graph. It's not moving at that stage, so we actually have a stalled tug-of-war at that moment. So some general characteristics of the model that I see are that there are two regimes of behavior. If I choose to model the motors in such a way that they detach easily, they have a low detachment force, so it doesn't take much for them to detach from the microtubule, then I get a regime of behavior where it's constantly switching back and forth. So this is a histogram that shows, over here this is the number of kinesins attached, over here is the number of diamines attached. So this histogram shows us the probability to have any combination of numbers attached. So what I can see from this graph is that we're really likely to either have on the order of 5 to 10 kinesins attached and 0 diamine, or several kinesins or several diamines attached and 0 kinesin. It's usually one way or the other. And that's because they can easily detach each other, so we get those cascades of detachment. And so we're in kind of a winner-takes-all scenario, where once one team starts to win, it takes over, everybody on the other side gets detached, and they take off in one direction. So this regime would be characterized by movement of the microtubule that goes a long way in one direction, very brief pauses, and occasionally would turn around and go a long way in the other direction. The other regime of behavior that I see with the model is that if I assume that the motors do not detach easily, they have a high detachment force, then we get more situations like this where we have a large number of motors of both types attached to the microtubule, and we end up with stalled tug-of-wars. So this regime of behavior would be characterized by long pauses interspersed with short movements and then more long pauses. So the point here is that the model can predict regimes of behavior that have distinct characteristics that we could then compare with the experimental data to see what's really going on. And like I said, we're at a very early stage of this project. They are working on getting more movies, and I'll be working on the computational side of it to predict what type of motion we expect under different conditions. And the features that we would like to compare to understand the system better include the velocity distribution, the frequency of reversals. So if their movies show us that the microtubule can change directions frequently, that tells us something about the underlying mechanism that would be different than if it always goes the same direction and very rarely reverses. We can also look at experiments where different motors are inhibited and see what happens in real life, what does the model predict. Okay, so I'd like to change gears now and tell you very briefly about one of my other areas of interest, which is on the cell scale, how do cells move? So, like I said, cell motility is very important for a variety of processes such as wound healing, and also cancer metastasis is another example where cell migration is in play. And this movie that I'll show you is one of the first direct observations of cell motility in a movie. This was taken in the 1950s. And what you'll be seeing is a white blood cell chasing an infectious bacteria. So to orient you, this little black speck here is the bacteria. This is the white blood cell. These spherical objects won't really be moving. These are the red blood cells in the bloodstream. And what we're going to see in this movie is that the white blood cell needs to catch this bacteria. Get it playing. So there they go. The white blood cell is turning corners, squeezing through barriers, even changing its shape in order to go after that bacteria. Got it.
The good guys win.
So who is everyone rooting for? So one other example that's shown over here is in axon outgrowth. So here's the cell body, here is the axon, and during nervous system development, the axons need to grow in a particular direction to reach their synaptic target. And that growth is led by a structure called a growth cone. And it moves by similar mechanisms as cell movement in order to lead that axon outgrowth. So I'll show you a quick movie of that process. So there's the cell body at the bottom. Here's the axon moving up. Led by the growth cone. So, if you think about a cell kind of like a vehicle, where it has some kind of transport mechanism and we want to understand how it moves, there are some elements that it would need to have. So, first, it needs to have some way that it's going to convert chemical stored energy into mechanical energy. It needs to exert force and be able to move. Another thing that it would need is traction. So when you're driving your car, you need the tires to have traction with the road. Well, the cell needs a way to have traction with the surface, but not so much traction that it's just stuck, right? And another thing that it would need is a steering mechanism. We saw this turning corners and actually following the bacteria, so there must be some mechanism for it to steer. So first let's talk about kind of the engine, or what's driving the motion. One hypothesis is commonly called the adhesion clutch hypothesis. And the way it works is, so in this schematic, we're looking at a bird's eye view of the cell. And near the leading edge, what this cartoon shows is a dense network of actin filaments. And these actin filaments are oriented in such a way that they tend to grow at the leading edge. They're polymerizing here. And further away, they are depolymerizing. So now if we kind of go to a side view of this system, we're looking at this actin network. And like I said, it's going to grow near the leading edge and shrink or depolymerize further away. Now, if this network is not adhered to the surface anyway, then think about what will happen as it grows on this end. It will grow, and then it kind of pushes up against the leading edge like a barrier, and Newton's third law says that that leading edge is pushing back on it. So it pushes that network away from the leading edge. And without any traction to counter that force, it's just going to slide away from the leading edge just as fast as it's growing. So this situation is sometimes referred to as treadmilling. And if we're thinking of our cell as a vehicle, this is kind of like the engine turning over, but you're not going anywhere. Then in this clutch analogy, when the actin network adheres to the surface, this is sometimes likened to engaging the clutch, because now, when the network grows at the leading edge, it's not slipping back anymore. So now this polymerization could push on that leading edge membrane and actually push it forward, allowing the cell to begin to protrude in the direction that it's trying to move. So this is a couple of projects that I'm not going to go into the details of, but what I want to tell you is that we developed mechanical models using similar approaches that I told you about in those previous projects, but to try and understand this actin adhesion clutch mechanism that I told you about in the last slide. So we developed a model where we tried to take into account what we thought were the important components mechanically, and we wrote equations of motion based on the forces acting on that actin network. And then we compared the predictions of our model with what was seen experimentally in different types of cells. So we worked with both Paul Forscher, who studies nerve growth cones, which, to remind you, are those structures that lead the outgrowth of axons. And we also worked with Margaret Gardell, who studies epithelial cells. And basically what we found was that this kind of simple clutch mechanism did a very good job at predicting or agreeing with some of the characteristics of their experimental observations in these two different systems. So some of the things that we were able to reproduce is the actin flow speed. When this actin network is moving in the frame of reference of the cell away from the leading edge, we were able to predict molecular distributions of things like myosin molecules that are embedded within the network and distributions of adhesion molecules. We were also able to accurately predict actin density distributions and spatial distributions of the traction. And the key point I want to make in mentioning these projects is to say that at this stage, we feel kind of comfortable that we have some idea about what's going on with sort of the engine clutch part of the system. We have some idea about how the vehicle moves, but at this stage in the field, I don't think there's a really good understanding of the steering mechanism. So how does it turn, and how does it know which way to turn? So this is another future direction that I'm interested in looking at, is trying to understand the steering mechanism. So in this schematic, we're looking at a growth cone, and in this picture here, the growth cone has a decision to make. So here we have some chemical cues that are known as chemorepellents. These are chemical signals that the growth cone should want to steer away from. And this is a chemoattractant. These are chemical signals that the growth cone ought to steer toward. And so a properly functioning growth cone in this scenario will come and it'll turn a corner and go toward the chemoattractant. And so we want to understand how it knows to do this. How is it taking a chemical signal and translating this into a mechanical response? And one kind of scintillating idea is that it may have to do with these microtubules. So, as I told you before, there is a bundle of microtubules in the axon, and occasionally these microtubules grow and extend into the leading edge of the growth cone. And they're undergoing a process called dynamic instability, where the microtubules are growing and shrinking, constantly moving. So these microtubules are actually exploring different regions of the growth cone as they grow and shrink. So what does this have to do with steering? Well, some people have performed experiments where they inhibited them, so they are no longer exploring that growth cone. And what they found was that the growth cone can still move, but it no longer steers properly. It becomes blind to these chemical signals that it ought to be following. So that suggests that this dynamic exploration of the growth cone by the microtubules is essential for the microtubules to be able to steer properly. So there are a few different hypotheses that we could look at to try and understand what exactly is going on there. How is the microtubule system interacting with that actin adhesion clutch that I told you about? One hypothesis that I'd be interested in testing is what I would call the asymmetric adhesion hypothesis. The idea is that if, say, there's some chemoattractant over on this side of the growth cone, and that triggers the actin network on that side to adhere a little more strongly than on the other side, then the microtubules that are exploring the whole growth cone begin to aggregate over at the site where there's more adhesion. And as they do that, then as we have more microtubules there, then materials can be delivered along those tracks, delivering signaling factors that would actually further promote the adhesion. And we think that there may actually be a positive feedback where adhesion in one region causes microtubules to aggregate. Aggregating microtubules causes the actin network to adhere more strongly, which in turn causes more microtubules to aggregate to this region. Now, what does this have to do with turning? Well, think about it. We have an asymmetry in the adhesion, so one side is adhering more strongly than the other. It's kind of analogous to, say, if you're pushing a shopping cart and one of the wheels is sort of stuck, and you try and push it and it's going to turn, right? Because you have one side that's stuck and one side that's going more quickly. So this would be kind of an analogous situation to that. So that's one hypothesis for what might be going on. And an ongoing project would be to develop a computational model where we incorporate both the growing and shrinking of the microtubules with the adhesion clutch system that the actin is a part of, and put those together and see what it predicts, and compare that with experiments. So one type of experiment that we would be comparing it with would be experimental measurements of the distributions of microtubules. And we could compare this model with experiments where microtubules are inhibited, experiments where actin is inhibited, or where myosin is inhibited. So that's another kind of future direction. And to conclude, I just want to say that these different projects are all examples, I think, of where computational modeling in collaboration with experiments can help us answer some interesting questions that are right at the boundary between physics and biology. So I would like to thank a variety of people, in particular some of the experimental collaborators that have played a really essential role. I've mentioned a few of them already. And I'd also like to thank Heiner Limke, my PhD advisor, and I would like to thank Alex Mulgilner and other members of the Mulgilner Lab at UC Davis, which is where I worked before coming here. And thank you to all of you for coming today, and I'd be happy to take questions.